right, good afternoon, and welcome to another edition of the Botany Bistro. Thank you so much for joining us. It is a chilly day here in Cincinnati, Ohio, but as you can see, it's nice and bright, sunny outside. And so we're still kind of edging towards winter, getting into our late fall season here. And most of the trees have lost their leaves. But some of them are still holding on for just a few more weeks. So my name's Mary, and I am the Ecology Education Manager here at the Civic Garden Center, which is in Cincinnati, Ohio. And we are doing this Botany Bistro series as a way to offer some bite-sized botany to anyone who is interested. You can join us live every other week on Fridays at noon. To get the joining information, you can visit our website at www.civicgardencenter.org. This is our 12th session, and so if you've missed any of our sessions, feel free to catch up. They're all archived on our website and also on our YouTube channel. We have covered things like systematics and taxonomy, plant anatomy, morphology, and plant physiology was our most recent couple of sessions. So we're actually starting a new topic today, and we're going to be talking about plant pathology. Now, I designed these sessions so that you can listen to them on your lunch break. You can run around and do errands and have it on in the background. So don't worry if you're not able to watch the screen the whole time. You'll still be able to get all of the information. At the end of the session, I'll turn on the chat box and we can have some questions and just enjoy some time with each other. So I just wanted to let you know that the Civic Garden Center has been here since 1942. So if you're local, please plan to come and visit us soon. There's always something new in the garden. And so it's a wonderful place to see the seasons change and just learn a little bit more about urban gardening and sustainability. I wanted to impart some of the botanical knowledge that I have learned over the course of my career and my education so that people can use some of these foundational biological concepts in their own lives. And understanding how plants grow and process things is a very important tool for gardeners, farmers, even if you've just got a couple of plants inside that you're taking care of. It's a really nice way to get that foundational knowledge so that you can identify issues and remedy them as quickly as possible. So we always have a little bit of a time to say what we're having for lunch, and my lunch tends to go along the lines of the topic that we're discussing today. And so I'm not going to give it all away. But one of my lunch items today are some grapes. So nice little sweet treat. And for my savory item, I have some purple potato soup. Smells so wonderful. There's some carrots in here as well. It's just delightfully seasoned and all farm fresh. So that is my lunch. I hope you're enjoying a healthy lunch as well. And so I'll tell you a little bit more about why I chose those items for my lunch as we chat today. So in our lesson, we're going to be learning a little bit about pathology and some significant events in history where plant diseases really became a problem. These were kind of global catastrophes. And so we'll talk about 5 of those. There are more that I'm not going to cover today, but I chose these 5. We'll be talking about tobacco mosaic virus, ergotism, wine blight, late blight, and tulip breaking. If you can stay with us on for all 5 of those adventures, that would be wonderful. But again, it is archived, so if you need to leave us early, that's okay too. We always have a little bit of homework. And so our homework from the bistro last time was talking about lenticels. And I posted my homework up on our Facebook page. So you can see some of the lenticels that I found in my yard. I found some really interesting lenticels on my pawpaw tree and on one of my pears. So I thought that that was a good share. Our homework for today is in the next couple of weeks, please tell us about a food ingredient that you would hate to lose to a pathogen. You're like, oh my gosh, I just couldn't live without this ingredient. And you can share that with us. All right, so let's see what is plant pathology. Now, this is a pretty big topic. Like I said, I'm just going to be covering some historical scenarios today. And in 2 weeks, we will continue the conversation. So on November 16th at noon, you can join us again. And I'll have a special guest with me, our Urban Agriculture Coordinator here at the Civic Garden Center, Kaimisha Montgomery, will be joining us. And we're going to be talking about plant diseases that we see commonly in our gardens and our farms, and ways that you can help prevent those diseases from overtaking some of your favorite plants and your crops. So be sure to tune in next time for that. That's going to be on November 16th. And we'll get really into the details of things that we see very frequently. Plant pathology, in a broad sense, is the study of plant disease, infection, and just a way that we can mitigate crop loss, make sure that we are doing what we can to maintain healthy agriculture, strong ecosystems. And the major causes of these diseases tend to be fungus, bacteria, viruses, and nematodes. So I'm not going to go into all of the specific details on each of those because that would take a long time. But those tend to be kind of the major players in plant pathology. In order for a plant to contract a disease due to fungus or bacteria or virus, it needs to have certain environmental conditions that are met. And so they kind of call this the triangle of disease. It depends on the host, the environment, and the pathogen. Understanding the life cycle of these diseases helps botanists and other scientists be able to create resistant strains. We'll talk a little bit about breeding when we get into biotechnology, which is not our focus for today, but will be the focus for a future session. And so thinking about how these disease vectors all kind of come together to form a perfect storm, if you will, for plants to become infected, we're really focusing on what are the environmental conditions, how is the disease being spread, and what is the role of the host plant in the disease itself. And so understanding all of those pieces is fairly complicated, and we really didn't have the tools until quite recently to be able to identify some of these pieces of the puzzle. So understanding how proteins work, RNAs, how things enter into plants. These viruses couldn't even be visually recognized until the development of very strong microscopy tools. And so it's not necessarily a new science, but we're learning so much about it every day. And so it's really exciting to be kind of on that cutting edge of research and figuring out how we can address some of the issues that we're seeing in some of our major crops. Making sure that the world has a secure food source is obviously very important for our survival. And so let's think about these 5 different stories that I'm going to tell you today. And we're going to start off with the tobacco mosaic virus. So this virus was one of the first that was ever identified and studied. And so today we know that viruses can be found everywhere: the air, the ocean, the soil. There are a small percent of viruses that are very dangerous. We've all been familiar with the coronavirus. And so causing worldwide pandemics is something that viruses are pretty good at. They're small, they spread easily. We do have, non-threatening viruses as well. Different viral pieces that kind of float around our lives and don't bother us as much. But the study of viruses did not start as medical science, but it started in the study of plants. And It took a long time for scientists that were studying these things to actually convince their colleagues that viruses existed at all, because they just weren't visible. They're much smaller than bacteria. They can't typically exist outside the host for a significant amount of time, so it was harder to identify them. But in the mid-1800s, this mosaic disease threatened tobacco crops, and plant pathologists needed to identify what was causing this, because really this is their livelihood. And so when you start to see the patches appear on your tobacco leaves, you're getting concerned that you're not going to have a good crop, and that could really upset your entire economic stability. So it was thought at first that it was bacteria or a fungus, something that they were a little bit more familiar with. Sometimes you can even identify, how that is being spread, whether it's from the infection from an aphid bite or another type of insect that is causing a wound on a plant. You can tell maybe if certain tools that you're using have been contaminated because the cuts that you've made on certain pruning plants have all kind of exhibited similar symptoms. But it was just a lot more tricky to identify what was going on with the tobacco mosaic virus. So these leaves are turning kind of a dark green, yellow, and gray. And it would cause up to 80% of crop loss in affected fields very, very quickly. And then they would replant the field the next year, and the same thing would happen again. And so they're, oh my gosh, how is this continuing to happen? And so once it reached a field, it really did spread rapidly, and successional plantings were continually battling this virus. So over a century ago, tobacco mosaic virus was the first virus to be discovered and the first virus ever purified. It's yielded some really neat insights about how viruses infect their hosts, and it's also led to Nobel Prize winning discoveries on principles of life and biology in general. And so other symptoms include leaf curling, stunting, yellowing of these plant tissues. The symptoms are dependent on the age of the infected plant, again, those environmental conditions, the strain of the virus, and the genetic background of the host plant. So there are different types of tobacco that are grown, and they are affected differently. The tobacco mosaic virus can also infect tomatoes. They are known to have distorted fruits or a poor yield. But thankfully, currently, tobacco losses due to this virus are less than 1% in many parts of the world because resistant strains have been created. And so that's very exciting that we're able to overcome some of these major challenges that we have with the tobacco mosaic virus. It's a good story to be able to be positive about. So the way that they found out that it's transmitted is if an infected leaf rubs against the leaf of a healthy plant. Tools that have not been properly cleaned can contaminate other plants. It can also contaminate seed coats, and so plants germinating from seeds can become infected. And so it's not even just the grown plant, it's also the seed. So it was really these early farmers noticing it had a lot stacked up against them in order to be able to control this. It's very stable, and it's been reported to be infectious even after 50 years of storage in a laboratory at 40 degrees Fahrenheit. So it doesn't die quickly. It's a movement protein to spread from cell to cell through the plasmodesmata. We did cover some cell structure, so if you want to refresh on plant cell structure and kind of get those terms down again, feel free to look at the archive. But that plasmodesmata connects plant cells, and typically they're too small to allow for passages of intact tobacco mosaic virus particles, but the movement protein enlarges the plasmodesmatal openings so that it can move between adjacent cells. Whoa. So that was a big discovery. Oh my gosh, like it's actually got, little friends inside that are helping it infect different parts of the cell. So it would move to adjacent cells and then release the protein and host proteins and initiate a new round of infection. So over and over, this cycle was happening. Eventually, this would reach the plant's vascular system, and once it gets into the veins of the plant, it has systemic spread throughout the plant using the phloem to get to the roots and the tips. So really not a good scene. But the tobacco mosaic virus disease cycle is completely dependent on the host for replication and spread. And so it would not necessarily be something that would be floating around in areas that don't have tobacco or tomato plants growing close together. But it can survive and overwinter, like I said, with those other testing that they've been doing. And so even infected plant debris or perennial weeds can be hosts, and also it can maintain in the soil. Because of the recognition of this early virus, there were some other breakthroughs that led to the establishment of virology as its own agent of science, its own discipline of science. And it's continued to be at the forefront of virology for the past century. So I just wanted to make sure we are all on the right page with our tobacco mosaic virus knowledge. But really neat stuff that botany has contributed to in the larger scientific world. The next topic I want to talk about is called ergotism. And we just wrapped up Halloween, and I hope that all of you had, some trick-or-treaters to visit, or just a nice piece of pumpkin pie, whatever your Halloween style is. But why would I say that plant pathology and Halloween are connected? Now, this is a little bit of a wives' tale. I guess urban legend. Some people do not agree that there is a connection here, and other people do. So I'll let you decide on your own. But let's talk a little bit about what ergotism is. So sprinkled throughout medieval European literature are reports of mysterious outbreaks that would leave thousands of people dead or disabled. Entire families would be afflicted with symptoms of like burning rashes, gangrene, convulsions, headaches, and hallucinations. And so it's called ergotism because it's a form of poisoning that comes from ingesting grains. Typically it's rye that have been infected with a fungus. And so this fungus replaces the regular healthy grains of the rye with dark, hard ergots, called ergos, that get mixed into the healthy grain during the harvest and the milling. And it was spread to the general populace just through the lack of attention to cleaning and making sure that these were being filtered out before they were being processed and kind of introduced into the mainstream of the food system at the time. And so the rye is a really robust grain that can withstand cooler temperatures and poor soils better than other grains. So it was widely planted, and still is today. It's a very important cover crop, and we use it a lot for our delicious breads. I also have heard this morning, my husband told me that there's a rye beer out there, and so we use rye a lot. It can be sown late in season and grow slowly over the cold months, and then really kind of takes off in the spring, growing flowers and ripening again in the early fall. So a rye infection begins when this airborne fungus spores infect the flowers of the grain and produces a sugary yellow slime that has additional spores and attracts insects, which then facilitate the transmission of this fungus to other flowers. Wind and rain and splashing can also spread these spores and transmit it to uninfected areas. So it spreads in a lot of different ways. These convulsions, muscle spasms, vomiting, hallucinations, and just pain in general on victims' toes, limbs, noses, and were said to say that they were eaten up by holy fire. So it doesn't sound pleasant at all to be dealing with this. Often victims would lose parts or entire pieces of limbs due to blood vessel constriction. And so it's said that Julius Caesar lost legions of soldiers to ergo poisoning during his campaigns, and that several severe epidemics in France between 900 AD and 1300 AD killed between 20,000 and 50,000 people. The ergo fungus contains a number of highly poisonous and psychoactive alkaloids, including LSD, which was synthesized from the ergovirus in 1938 by chemist Albert Hofmann. So occasionally these things get studied and continue to find out different things for them to be used in different areas. But the reason why I kind of mentioned Halloween at the beginning of this story is that it is a hypothesis of some that the Salem witch trials was actually a result of ergo poisoning. And so there were people in the community that were having seizures, convulsions, hallucinations, and these translated into them accusing each other of being witches. Now, some of this has been just questioned or debunked, but how interesting that is even a concept that could be possible, that it could have been a plant pathogen, a plant disease, that then led to these very bizarre, strange trials that we still remember today. It's typically rare because there's a lot of screening that happens now, so that when grains are brought in for milling, they're able to be identified and they're able to be sorted out. So you don't need to necessarily be concerned about this type of poisoning now, but definitely it plagued our ancestors widely. All right, so now we're going to move on to one of my lunch items. As much as I would like to also be enjoying a glass of wine with my lunch today on the bistro, I'm very happy with my grapes. And so this next disease we're going to discuss is called the Great French Wine Blight. So this was a very tragic situation that happened as the result of improvements to transportation systems and infrastructure. So something that was seen as very good kind of had a pretty bad end result. So phylloxera was introduced to Europe in the late 1850s. The blight caused by this lice or louse annihilated over 2.5 million hectares of vineyards and also caused riots in Burgundy. There were soldiers that were guarding vineyards. I mean, it was like super serious. People were thinking that it was a sign of the devil. You know, it was just all over the place. Why are all of these vineyards just suddenly dying after having been established for so many generations? And so this phylloxera is a tiny little critter, and the females are barely visible to the naked eye, and they feed off of the roots. The phylloxera will kind of penetrate the root and create a scab or a scar, a wound, and in there they'll kind of suck out the nourishment that they want. Maybe getting some of the sugars in there. And then they infect the root with a poisonous secretion that keeps it from healing so that the wound stays open. Who doesn't love a 24-hour buffet? So the phylloxera essentially set that up for themselves. And so the wounds could not heal. Eventually, this poison does kill the vine. And the females continue to lay eggs. And crawlers that move to other roots, and so the disease kind of fans out in a circular form around the original infection site. So, why was this happening so quickly? It really kind of shocked people because grapevines had been traded between the Americas and Europe for over centuries. And what happened was the advancement in steamships with improved engines that made the trip much shorter than a wooden sailing ship, which usually took many months, enabled the phylloxera to survive the trip. And so with that decreased travel time, you had an increase in the fertility of these pests. And so today it's still spread by rootlings or dormant plants and can be in the soil. And so as equipment moves through the soil, or people that are working in the fields, tracking it around on their shoes, they spread very well. So we've got this phylloxera lice just kind of running all over, running rampant. What they found out to do in order to save the wonderful wine production that happens in Europe and France, other areas, was that they could graft French vines onto American rootstocks. So American rootstocks are able to form a scab over this penetration site, and the European rootstocks could not. So the vines of American rootstocks don't bleed to death and are able to survive with this pest. It took a while to figure out that this would work. Hybrid plants did not always work because sometimes the leaves could still become infected. But this little tricky phylloxera had a couple of different adaptations that really made it challenging for people to figure out. It has a sexual stage, but can also ignore that and can reproduce asexually. So it took a while to kind of figure out what they could do. But grafting vines onto American rootstock was a great fix so that the soil that is infected and the roots that are infected are able to withstand that infection, and the stock that is grafted is still true to those traditional old world vine species. And so you get those wonderful traditional wines that we all appreciate, and you're not having to really do a lot of compromising on changing the entire species that you're growing, the entire variety that you're growing, so that this rootstock was able to be used. And so actually it took a while to convince the French to do this. Burgundy outlawed this idea of grafting until 1887. And for about 20 years just would not allow it to be part of agriculture in the wine industry. And then finally they were like, okay, there's not a lot else we can do. But there are a lot of wines that we enjoy from Europe that are grafted onto American rootstock. So that's a nice little fun trivia you can talk about at your next party. All right, I've got two more pathology stories for you today. And so thanks for hanging on, everybody. The next one is late blight. And so this one is responsible for the Great Potato Famine. And it's actually a reason why my grandmother and her family moved here from Ireland. And so I am a result. My American citizenship is a result of this. And so the humble potato is native to the Andes and was cultivated by the Incas. It had been cultivated for a long time. They domesticated hundreds of types of potatoes. So here, enter in my purple potato soup, which is just one of many, many varieties of potatoes. So I do encourage you to go out there and try new types of potatoes if you haven't. But the invading Spaniards only brought a handful of varieties back with them to Europe during the second half of the 16th century. And by the end of the 17th century, only one or two of these established varieties were being grown widely. And so really they had a monoculture of just a couple types of potatoes. it became a major crop in Ireland because these soils were poor and people had struggled to support themselves with traditional cereal grains. So the potatoes were very instrumental in becoming a staple crop for them to survive. And so it's propagated by tubers rather than seeds. And so people were passing around these tubers, but what that resulted in was genetically identical potato plants. And it's estimated by the early 1840s that half the Irish population had come to depend almost exclusively on potatoes for their diet. So I do enjoy potatoes, but I'm not sure if I would want them all the time. That's why we have so many different ways to eat potatoes, I suppose. But then came this fungus, and it's similar to a water mold, and it has the ability to decimate potato plants with a disease known as late blight, introduced to Ireland from North America in 1845. Late blight thrives in wet conditions, and unfortunately for Ireland, the summer of 1845 was a very wet one. Much of the crops that year rotted in the fields, and the disease became established in the areas where people were growing. There was almost no genetic diversity, and so this mold just kept spreading and spreading and spreading because it found an endless buffet that it could eat, and so it really just wiped out entire communities' food sources. And so the devastating failures between 1846 to 1849, caused starvation and other types of terrible famine-related disasters. There were no potatoes to eat or sell. You couldn't pay rent to your landlord. People got evicted and sent to workhouses. About 2 million Irish immigrated during the famine. And many of whom came to North America, and of those that stayed, it's estimated that about 1 million of them died from starvation or typhus or other famine-related diseases. So big deals. What does it look like? Let's see. Late blight infects leaves, stems, and fruit. The leaves will have large, dark brown splotches on them with kind of a green-gray edge. And then the infections progress through the leaflets and the petioles. And so you'll see large sections of dry brown foliage. It's not something that is easy to miss. You see this one right away. The cool, wet weather really helps spread this fungus. And it can produce thousands of different spores in less than 5 days. They become airborne and are just unstoppable, basically. And so what do we do now? We have resistant varieties. We try to make sure that we have good ventilation around plants, but also it's that diversity piece, making sure that we're not reliant on just one variety so that we have genetic diversity that creates a stronger defense against pests and pathogens. All right, so we've got one more historical story to tell, and then I'll open up the chat for anyone who might have questions or want to share something about their experiences. And so the last one is the tulip breaking virus. So tulips were introduced to Europe from Turkey shortly after 1550. And in the early 1600s, the demand for unusual tulips among the wealthy began to exceed supply. Prices for these bulbs climbed and climbed. And so it was interesting, this tulip mania kind of developed in the Netherlands, and they were growing lots of different varieties, but it was this one particular one that really started to gain a lot of popularity, and it was very unique. We'll talk a little bit more about that one. So there was this peak of tulip mania between 1633 and 1637. a lot of people started to get into the market, into speculating. And so middle class and poor families were mortgaging their homes and businesses so that they could buy and resell bulbs at higher prices. These were largely based on contracts, and so sales and resales could be made many times over without either the seller or the buyer even seeing the bulbs. But this one particularly rare one, known as Semper Augustus, was streaked. It had white and red and is believed to have been the most expensive tulip in the frenzy, and is also painted in several Dutch floral masterpieces. And so this is called a breaking. And so broken tulips are very different from regular tulips. Typically tulips will have a solid color to their petals. Maybe they'll have like a dark patch or kind of a nectar guide towards the center where the pollen and the nectar are stored. But typically those petals are deeply colored. And so these broken tulips with these streaks were just so different. People really gravitated towards them. And so today these legendary breeds don't exist. You cannot find a Semper Augustus right now. And let's think about why. So they've actually become illegal in parts of the Netherlands because they don't want them to be integrated with their other tulip crops. And so when the crash came early in 1637, many speculators could not even afford the cheapest bulbs, and they were like, this is getting out of hand. And so almost overnight, the price structure for tulips collapsed, leaving many ordinary Dutch families in financial ruin. So not a good thing for some families. And the common solid color tulips that we have now are developed through traditional breeding methods. But most of these broken strains that fueled this mania have died out because the virus that gave them this very unique pattern also made them weak. And so what happens is that the bulbs are smaller and their vitality is less. You can tell the petals kind of droop a little bit. They're not quite fully formed sometimes. And so it was, yes, you have this streaked, petal variety, but it doesn't always come true. And so that means that you couldn't collect more and more and more bulbs like you could for some of the other varieties because they just were continually smaller and more deformed and less fertile when you tried to plant them. And so the rarity that was caused by this bulb being not being able to be propagated easily also increased the price, but then everything just kind of crashed down, like I said, overnight. And so they found that this virus that was causing the breaking is spread by aphids. It infects the bulb, and the primary color of the tulip is suppressed, leaving the underlying white or yellow to show through. There are different hypotheses on exactly what symptoms there are, and they can vary based on the type of tulip, the strain of the virus, the time of infection. Remember that triangle we kind of talked about at the beginning of our session today that mentioned, that you need to have the environment, the pathogen, and the host kind of all in the same cycle for this to be something that really is becoming a problem. And so over time, the virus weakens the bulb, and then its daughter offshoots are also infected with the virus. With each new generation, the bulb grows weaker and weaker until it has no strength left to flower and withers away. And so that is why Semper Augustus has now gone extinct. So I'm going to go ahead and say thank you for attending today. I'm very happy that we were able to kick off plant pathology together. I'm going to go ahead and open up the chat for anyone that would like to share questions or just ideas. And remember to tune in next time, which is going to be the 16th at noon, when you can learn about some common diseases that we see in our gardens and get some remedies for those, so that we make sure that we're all tending our plants the best that we can.